All right, so in this video, we're going to look at one type of membrane transport. Really specifically, we're going to look at passive transport. But let's just look at the membrane properties of why you need to have transporters. And really, the answer is because of this phospholipid bilayer, you end up with this section here that is hydrophobic. So you have a hydrophobic core. And because of that, nonpolar things, so nonpolars cross easily in and out of the membrane, not a problem, okay? Which is fine, and small polar molecules also can go through because they're so tight, okay? So they're small enough to go through. So think like water is actually small enough to go in and out. The issue is large polar molecules, like big, like glucose or sucrose, any of the sugars really, and ions, so things like sodium and potassium and all that kind of stuff. They cannot pass without some kind of assistance, and so we're going to see what that assistance is in a little bit. But let's talk about how really all of these guys can get through, and that's really with this process of passive transport. Very few things actually need active transport. So let's passive transport. The key to it, the key thing is it does not require cellular energy. And if you're asking yourself what is cellular energy, you should be answering ATP, okay? So it does not require ATP, it does not require cellular energy for it to get through. Okay. This is getting more tilted. Okay. So does not require cellular energy, key part. And what happens is molecules are just moving around, and molecules move what's called down their concentration gradient, or they move from where they're highly concentrated to where they're less concentrated. Okay, so they move down their concentration gradient. So let's look at this picture. This is simple diffusion. So if you have these black molecules and they're kind of isolated, they're pretty highly concentrated there. And if we let them go, they're going to start to move around randomly until they're evenly distributed. Okay, so we have this random movement of music. We have a random movement of molecules down the concentration gradient, from high concentration to low concentration. Any molecule is going to move from where it's highly concentrated to where it's less concentrated until an equilibrium is reached. And that equilibrium is going to continue where there's some molecules are going to go in and some molecules are going to go out, but at the end, the same amount of molecules are going to be inside as outside. Okay, so let's look at a very specific type of diffusion, which is the process of, or the idea of osmosis. So osmosis has to do with water, okay? So it's the diffusion of water across a selectively permeable membrane. And we're going to look at this in lab. So it's the diffusion of water across, I'm just going to write, a membrane. And what ends up happening is you end up with three kind of cell conditions. The first cell conditions is isotonic, or you can even have solutions, okay? Where the two solutions have the same solute concentration. I'm going to start abbreviating concentration like that. Okay? Hypotonic is where the solutions, when you're in a hypertonic situation, is the solution has a lower solute concentration. We'll look at some examples here in a second to help make this sense. And then a hypertonic is the solution with a higher solute concentration. Okay. So we're going to look at animal cells and plant cells, and they kind of do different things. In an isotonic situation, because the solutions are the same, no water movement, or no net movement of water. Okay, water's going to move in and out, but there's no net movement, no water movement. Okay, This is normal for animal cells. This is where they want to hang out, okay? So no water movement or no net movement of water in and out of the cell. In a plant cell, when it's in its isotonic state, again, there's no net water movement. But this cell is what's called flaccid, or it's like limp, and it's kind of a sad little cell, okay? And you can picture, like, if you're actually looking at the actual plant, it's wilted. Now let's look at what happens if you put them in a hypotonic solution. And so the environment is hypotonic. Okay, so there's hypotonic. Okay, and same over here. We have an environment that is hypotonic. So what happens is the solution is lower with solute concentration. So what happens is water goes rushing into the cell. Water enters the cell because there's more solute inside the cell than outside the cell. So water goes rushing in. More solute equals less water. That's the key part. And the cell swells and bursts or lyses. So the cell bursts. Can't spell burst. Let me try that. The key part is more solute, you have to remember this, more solute equals less water. So in a hypotonic solution, there's more solutes inside, so there's less water, so then this water goes rushing in to the point that it actually causes the cell to burst. Okay. Now with a plant cell, it's something different happens. The water is going to enter the cell just the same because of the same situation. There's more solutes, so there's less water, so water goes rushing in from the outside. But guess what? There's a cell wall that's going to push back, and so the cell becomes what's called turgid. This is where this is normal for a plant. Okay, this is its normal state. This is where it likes to be. It likes to be in a hypotonic situation. Now let's look at what the plants, neither plants nor animals like, which is an environment that is hypertonic. Okay. In this case, there's less solutes inside the cell and more solutes outside the cell. So water is going to go out of the cell. So water exits the cell. The cell shrinks, and you get what's called plasmolysis. And so the plasma membrane, as you can see, kind of pulls in, and usually the cell can end up dying. And look what happens in the poor plant cell with its cell wall, okay? In that environment that is hypertonic, okay, water is going to exit the cell just the same. And so, again, the cell membrane is going to pull from the cell wall, and we're going to end up with that plasmolysis condition, not a happy cell. Usually it can actually cause cell death, okay? So that's the different states of osmosis. And then, now let's look at how some of these animals, because if you live in a saltwater condition or if you live in a freshwater condition, there's some animals and organisms that have to deal with this. So this is osmoregulation. And this we're going to see over and over again. So in this case, the paramecium, it's in a hypotonic environment because it lives in freshwater, right? So there's more solutes inside the cell than outside. So if you think about it, water enters. And what happens is they have a contractile vacuole that actually pumps water out. That's how they adapt to that situation. So contractile vacuole that pumps water out. Now what if you're a freshwater fish? You're in the same kind of situation. You're in that hypotonic environment, okay? And so water's going to enter. So what they do is the way they deal with it is they actually don't drink anything. They have no need to drink water like we do, okay? And their urine is extremely watery, which works out. I mean, they live in the ocean. It's not like they need to deal with the fact that they have to excrete it all the time. So urine is extremely watery. Okay. And then marine bony fish, like this cute little seahorse here, they live in a hypertonic environment. This is why you're like, you lose a lot of water when you go in the ocean, okay? So what they do is a lot of them drink lots of water, and their gills actually have lots of salt pumps to pump out water, okay? So gills pump excess water. Okay, so that's how water goes in and out. And a lot of times it goes through a carrier protein called an aquaporin. Now, what if you have not a lot of a solution in your cell, like a chemical in your cell, and you want to get it in, but you don't want to actively pump it in? That's where facilitated diffusion comes in. So you need what's called a protein to get it in, okay? So the facilitated diffusion it's still part of the passive diffusion process because there's no energy that's required, but it's diffusion of solutes across a membrane. So it's the same idea with help from a transport protein, an integral protein. That's the key part. Okay, it's an example of passive transport. And notice that there's high concentrations of the solute on this side and low on this side. So this one happens to be a carrier protein, where when the solute pumps in, it lands in this spot. It's specific because it's a protein, so it has a specific three-dimensional shape. And then when it goes in, it makes the protein change its shape. It's called a conformational change. And then it allows it to release on the inside where there's less of it. Okay. The key part about transport proteins is they are very specific. So you would have one for glucose, you would have one for hydrogen, you would have one for sodium, whatever they are. They're very specific to the specific solute that they're bringing in. That's the key part. Okay, so they're very specific. Transport proteins are specific. And so a lot of times they have two conformations or two shapes. One where they're open to the outside, and then one where they release it to the inside, okay? You can also have this facilitated diffusion, same thing. Instead of having a carrier that changes shapes, you can also have a channel. And so a channel protein, I'm just going to attempt to draw one. They're really not that exciting. It's what they usually look like this, and they just kind of have a big channel or chute that something can go through. Water actually a lot of times enters the cell through a channel protein, okay? So like if there's water out here, it goes in like that. So it just goes, it's like a big chute. Okay, so it's another example of a facilitated diffusion example. And that's all of our passive transport examples.